The search for extraterrestrial life represents one of humanity's most profound scientific endeavors. As our observational capabilities advance, astrobiologists have developed increasingly sophisticated methods to detect potential signs of life on worlds beyond our solar system. This process involves identifying biosignatures, observable evidence that strongly suggests the presence of past or present biological activity within the context of an exoplanet's overall environment. The detection of such signatures requires multidisciplinary approaches spanning astronomy, planetary science, biology, and chemistry, along with advanced instruments capable of gathering the necessary data from vast cosmic distances. Before astrobiologists can search for signs of life on distant planets, they must first detect these worlds using various observational techniques. Until around 2012, the radial velocity method, also known as Doppler spectroscopy, dominated the field of exoplanet detection. This technique measures the slight wobble of a star caused by the gravitational pull of orbiting planets. The radial velocity signal remains distance independent, but requires high signal-to-noise ratio spectra to achieve precision. Consequently, this method is generally limited to relatively nearby stars, approximately 160 light-years from Earth, particularly when searching for lower-mass planets. Despite these limitations, radial velocity observations can detect massive Jupiter-like planets orbiting up to 10 astronomical units away from their host stars, though such detections require many years of observation. The transit method, which became predominant after 2012 with the launch of NASA's Kepler Space Telescope, observes the slight dimming of a star's light as a planet passes in front of it from our perspective. This approach has proven remarkably successful, with Kepler and subsequent missions discovering thousands of exoplanets. The transit method works particularly well for large planets orbiting close to their stars, as they block more light and transit more frequently. When planets have high albedo, reflectivity, and orbit relatively luminous stars, their light variations become easier to detect in visible wavelengths, while darker planets or those around cooler stars are more readily observable in infrared light. Gravitational microlensing offers another powerful approach to exoplanet discovery. This phenomenon occurs when a foreground star's gravitational field acts as a lens, magnifying the light from a distant background star. If the lensing star hosts a planet, that planet's gravitational influence creates a detectable contribution to the overall lensing effect. First proposed for exoplanet detection by astronomers Shudi Mao and Bohdan Podzinski in 1991, and refined by Andy Gould and Abraham Loeb in 1992, microlensing became practically viable in 2002 through the optical gravitational lensing experiment, OGI. This method's significance lies in its sensitivity. It was the first technique capable of detecting Earth-mass planets orbiting ordinary main-sequence stars. The pulsar timing method detects planets by measuring anomalies in the extremely regular radio pulses emitted by pulsars, rapidly rotating neutron stars. Like normal stars, pulsars move in small orbits if they host planets, creating measurable variations in pulse timing. While not originally developed for planet detection, this method can identify remarkably small worlds, less than 1/10 Earth's mass, and can even reveal gravitational interactions between multiple planets in a system. In 1992, Alexander Wolfsjohn and Dale Frail used this technique to discover planets around pulsar PSR 1257+12, providing the first confirmed detection of planets beyond our solar system. However, the method has limitations. Pulsars are relatively rare, special circumstances are required for planets to form around them, and the intense radiation near pulsars makes these systems unlikely abodes for life. Several other detection methods complement these primary techniques. Relativistic beaming, also called Doppler beaming or boosting, first proposed by Abraham Loeb and Scott Gaudy in 2003, measures changes in a star's apparent brightness caused by its motion due to a planet's gravitational pull. While this effect is subtle, barely detectable even for a Jupiter-mass planet orbiting very close to a Sun-like star, it provides valuable data without requiring precise stellar spectra, making it useful for studying fast-rotating or distant stars. The first planet discovered through this method, Kepler 76b, was announced in 2013. Once exoplanets are detected, astrobiologists turn their attention to searching for potential signs of life. Biosignatures are substances or phenomena that provide scientific evidence of past or present life. In the context of distant worlds, these primarily consist of gases and other substances, including pigments, that can produce distinct spectral or photometric signatures observable with telescopes. These chemical markers must be interpreted within the broader context of the exoplanet's overall environment to distinguish genuine biological origins from abiotic, non-biological processes. The search for biosignatures relies heavily on the concept of biogeochemical exo-Earth system models that simulate potential biosignatures as they would appear in spectroscopic or photometric observations. Given actual telescope data, these simulations help calculate Bayesian likelihoods, statistical measures that quantify the probability of those observations occurring under different scenarios, both with and without life present. This approach explicitly accounts for false positives, wherein non-biological processes mimic apparent biosignatures. Oxygen stands as one of the most compelling potential biosignatures in exoplanet atmospheres. On Earth, atmospheric oxygen results primarily from photosynthesis by microbes and plants. The Exoplanet Biosignatures Workshop Without Walls, EBWWW, held in 2016, identified oxygen as warranting special attention, leading to an in-depth review that rigorously examined the nuances of false positives and false negatives when using oxygen as evidence of life. This analysis highlighted the need for contextual knowledge to properly assess claims regarding life on exoplanets. Beyond oxygen, several other atmospheric gases serve as important biosignature candidates. Methane, water, and carbon dioxide have strong spectral signatures that can be measured simultaneously in spatial footprints examined for biosignatures. These additional markers help identify possible correlations between biosignatures and water, while assisting in quantifying mixing ratios for trace gases by correcting for topographic and temperature effects using carbon dioxide measurements. The deuterium to hydrogen (DH) ratio offers particular potential for identifying active vents and discriminating between near-surface and subpermafrost release zones, providing crucial information about the depth of potential biosignature release that helps distinguish between geochemical and biological generation mechanisms. The detection of biosignatures on distant worlds depends critically on advanced telescope technologies. NASA missions like the Kepler Space Telescope have already documented thousands of exoplanets orbiting other stars, while the Transiting Exoplanet Survey Satellite, TSS, continues to expand this catalog significantly. These discovery missions identify promising candidates, particularly Earth-sized rocky planets orbiting in their stars' habitable zones, where conditions might permit liquid water to exist on their surfaces. The James Webb Space Telescope, JWST, scheduled to launch in 2021, represents a monumental advance in our ability to characterize exoplanet atmospheres. Building upon and extending the discoveries of the Hubble Space Telescope, JWST observes at infrared wavelengths, enabling scientists to determine whether rocky exoplanets contain oxygen in their atmospheres. The telescope's mid-infrared instrument, MIRI, offers particularly powerful capabilities for studying exoplanet atmospheres. Researchers have developed innovative methods to maximize the capabilities of instruments like JWST's MIRI for biosignature detection. One such approach focuses on studying the atmospheres of exoplanets orbiting nearby white dwarf stars, building upon the established IR-access method. This technique could help identify bodies in a star's habitable zone, including super-Earths, Earth-sized planets, and hot Mercury analogs. Calculations suggest that super-Earths in the habitable zone could be detected up to 10 parsecs from their host star using just 10 hours of observation time. More importantly, follow-up characterization of a planet's atmosphere to search for potential biosignatures would require only 20-35 hours of observation time. Some researchers believe that observing nearby white dwarf systems using this technique might provide the best opportunity to search for signatures of life using JWST. The next generation of advanced telescopes promises to sharpen the hunt for potential extraterrestrial life through increasingly detailed examination of exoplanet atmospheres. A 2024 study published in the Astronomical Journal assessed upcoming telescopes' ability to detect chemical traces of oxygen, carbon dioxide, methane, and water on 10 rocky exoplanets. The results indicated that for nearby worlds like Proxima Centauri b and GJ887b, these instruments would excel at detecting potential biosignatures, though only Proxima Centauri b would allow for carbon dioxide detection if present. While no exoplanet precisely matches Earth's early conditions for life, this research suggests that super-Earths, planets more massive than Earth but smaller than Neptune, could make suitable targets for future biosignature investigations. The interpretation of potential biosignatures requires robust analytical frameworks. The scientific community has developed comprehensive approaches to organize qualitative understanding and available data into formal quantitative structures, particularly using Bayesian statistical methods. This framework allows researchers to combine prior knowledge of factors influencing planetary habitability with observational likelihoods to calculate the posterior probability of life existing on a given exoplanet. Four essential components characterize this analytical process. First, researchers need thorough characterization of the stellar environment, including previous observations that provide context for interpreting biosignature data. Second, they simulate potential biosignatures using biogeochemical models of exoplanet systems. Third, they calculate Bayesian likelihoods by comparing actual observations with these simulations. Finally, they combine these likelihoods with prior knowledge to determine the posterior probability of life's presence. A crucial challenge in biosignature detection involves distinguishing true biological origins from abiotic processes that might produce similar signatures. Not all exoplanets resemble Earth, and even on planets that appear superficially Earth-like, oxygen might result from non-biological processes. Therefore, researchers must carefully consider the specific planetary context when interpreting potential biosignatures. To address this challenge, interdisciplinary teams have developed frameworks like the Detectability Index to help scientists narrow their focus to planets where oxygen most likely indicates biological activity. This approach acknowledges that while oxygen in Earth's atmosphere results from photosynthesis, its presence on other worlds might stem from entirely different mechanisms. By systematically analyzing planetary characteristics, scientists can identify the subset of exoplanets where atmospheric oxygen most strongly suggests biological origins. Despite significant technological advances, detecting biosignatures on distant planets presents immense challenges. The primary difficulty lies in the extreme distances involved. Even the nearest exoplanets are several light years away, meaning that the light we receive from them is extremely faint. Furthermore, separating the planet's spectral signal from that of its much brighter host star requires sophisticated observational techniques and instruments. Many detection methods also face specific limitations. The radial velocity method struggles with fast-rotating stars because their spectral lines become less distinct as half the star rotates away from observers while the other half approaches. The transit method only works for planets whose orbits align with our line of sight, severely limiting the sample of planets we can study this way. Relativistic beaming suffers from the small magnitude of its effect. The light variation is minimal even for large planets orbiting very close to their stars. Even when biosignature gases are successfully detected in an exoplanet's atmosphere, interpreting their origins presents additional challenges. Numerous non-biological processes can produce substances that mimic biosignatures, creating false positives that might mislead researchers into identifying life where none exists. For instance, oxygen can accumulate in a planet's atmosphere through photochemical processes involving water vapor, particularly if hydrogen easily escapes into space. The problem extends beyond individual gases to their combinations and concentrations. Researchers must look for patterns of multiple biosignatures and consider them within the planet's broader environmental context. A thorough understanding of a planet's geological activity, atmospheric circulation, and stellar environment is essential for properly interpreting potential biosignatures. The future of biosignature detection looks promising as new telescopes and instruments come online. Following JWST, other planned space-based observatories will offer even greater capabilities for characterizing exoplanet atmospheres. These instruments will provide higher spectral resolution and sensitivity, allowing researchers to detect fainter signals and distinguish more subtle spectral features associated with potential biosignatures. ground-based observatories also continue to advance, with extremely large telescopes, ELTs, under construction that will feature primary mirrors spanning 30 meters or more. These facilities, equipped with advanced adaptive optics systems to compensate for atmospheric distortion, will complement space-based observations by providing higher resolution data on nearby exoplanet systems. While current biosignature research focuses heavily on atmospheric gases like oxygen and methane, future work will likely expand to include a broader range of potential life indicators. Seasonal variations in atmospheric composition, surface reflectance patterns that might indicate vegetation or other organisms, and even technosignatures suggesting intelligent activity represent promising avenues for investigation. The scientific community continues to refine analytical frameworks for assessing potential biosignatures, developing more sophisticated models that account for the wide diversity of planetary environments and possible forms of life. These frameworks increasingly incorporate interdisciplinary perspectives, drawing on expertise from astronomy, planetary science, geology, chemistry, and biology to provide more nuanced interpretations of observational data. The search for signs of life on distant planets represents one of science's greatest challenges and most profound pursuits. Current methods combine advanced observational technologies with sophisticated analytical frameworks to detect and interpret potential biosignatures within their planetary contexts. While no definitive evidence of extraterrestrial life has yet been found, the rapid advancement of exoplanet science continues to expand our capabilities and refine our approaches. Future discoveries will depend on continued technological innovation, particularly in telescope design and instrumentation. Equally important will be theoretical advances in understanding the range of possible biosignatures and their abiotic mimics across diverse planetary environments. The ultimate goal, finding evidence of life beyond Earth, requires patience, methodological rigor, and interdisciplinary collaboration. As our catalog of characterized exoplanets grows, particularly those resembling Earth in key aspects, so too does the possibility of that momentous discovery. Whether life proves common or rare in the cosmos, the search itself continues to deepen our understanding of our place in the universe and the conditions that make life possible. Through this pursuit, we gain insight not only into potential extraterrestrial biospheres, but also into the precious uniqueness of our own living world.